So very good morning, good afternoon, good evening everyone. So we are discussing human anatomy and physiology, and in unit 3 we are going to discuss about blood clotting. We are discussing about blood. So how blood clotting actually happen and how blood clots, what are the mechanism behind the blood clotting. So that particular information we are going to discuss today. So blood clotting, what happens? If you remember, I told you in your positive feedback systems, why first question should arise in your mind, why blood in the systemic circulations, why it doesn't clot even though it has a platelets. So why it doesn't clot? Because our healthy endothelial cells are releasing some substances. This is your blood vessels and it's lined by endothelial cells, and these healthy endothelial cells are releasing some chemical substances which inhibit the attachment of platelet to the blood vessels. So these substances are your nitric oxide, these substances are your PGI2, that is the prostacyclins, their substances are your ADP dephosphatase. Whenever any injury happens, so in injury what will happen? Your blood vessel is going to damage. So damaged blood vessel, do you think these damaged blood vessels will release these substances? No. Of course, as I said, only healthy endothelial cells are releasing these substances, and these substances are inhibiting the attachment of platelet to the blood vessels walls. So when there is an injury, so what will happen? These chemicals will not release and these platelets is going to settle down, and this is the first steps that we call platelet adhesions. Platelet adhesions. And if you remember, I told you damaged endothelium is releasing one factor that is we called VWF, that is the von Willebrand factors, and with von Willebrand factors is particularly this is a factor which is released by the damaged endothelium, not in normal endothelium. So these platelet is going to adhere. So first step is platelet adhesions, and after adhesions these platelets are going to activate, and during the activations there are a lot of reactions which are happen in the platelet, like there is activations of phospholipases, and phospholipases will do the degradations of phospholipid, arachidonic acid will form, and there is a formation of TXA2. So TXA2 on platelet, it's a strong aggregator of platelets. So when these chemicals will be expressed on the surfaces of platelet during the activations, so what will happen? This chemical is the strong aggregator of platelets. So more platelets are going to aggregate. There are other substances which is going to release during the platelet activation steps, that is the ADP. ADP is also a strong aggregator of platelets. So all these chemicals, what will happen? Platelets will be aggregate much more here on the injured area. All the platelet is going to aggregate, and this forms a plug that is we call primary plug, and your bleeding going to stop, right? But now this primary plug is going to get the strengthened by the clotting factors. So that is this is a step from where your mechanism of blood clotting is going to start. So your blood will stop by the primary plug, but this primary plug is very loose plug. If you are doing some disturbances to the plug, again blood will be come out, right? So to get the strengthen to the primary plug, there is activations of blood clotting mechanisms. So blood clotting. So blood clotting happens. There are two mechanisms of blood clotting: intrinsic mechanism and extrinsic mechanism. And ultimately, finally, what happens? Your soluble fibrinogen will convert into the fibrin which are insoluble, and these fibrins will deposit on these platelets and get the strengthen. So you know, जब भी आपको कोई चोट लगती है उसके बाद दो तीन दिन बाद वो जो आपका क्लॉट हुआ था वो बहुत हार्ड हो जाता है. तो हार्डनेस कौन प्रोवाइड कर रहा है? फाइब्रिन. फाइब्रिन कैसे बन रहा है? फाइब्रिनोजन इज गोइंग टू कन्वर्ट इनटू द फाइब्रिन. दिस इज द लास्ट स्टेप्स ऑफ योर ब्लड क्लॉटिंग. So one by one we are going to discuss what actually the blood clotting, what its mechanism. So blood clotting's mechanism is, blood clotting, there are two mechanisms. One is intrinsic mechanism, another one is extrinsic mechanism. So what is the difference between intrinsic and extrinsic? Of course, as its name indicate, intrinsic, so means there are blood has intrinsic properties for the clotting. So you know, to understand this in a simpler concept, if you took the blood and put into a clean tube, only tube which does not contain anything, you took the blood and put into the tube, so what will happen? This blood is going to clot after some time, right? So what does it mean? It means blood has some intrinsic properties which helps in the clotting. आपने blood लिया, blood को एक ट्यूब में डाल दिया, तो थोड़ी देर बाद क्या होगा, blood clot हो जाएगा. क्यों clot होगा? Means blood has some intrinsic mechanism which is helping in the clotting of the blood, right? So that is the intrinsic mechanism. Extrinsic means, for example, you take the tubes, and in the tubes you have added, and this is the tube which contains the anticoagulant like EDTA, like citric acids, right? So now what you have taken, what you have done, you took the blood, but now in this case you took the blood in the tube which contains already the anticoagulant. So what will happen? If you took the blood and put into the anticoagulant tube, so do you think it will clot? No, it will not clot. क्यों नहीं क्लॉट करेगा? क्योंकि ऑलरेडी आपकी ट्यूब में एंटीकोगुलेंट है जो कि उसको कोगुलेट नहीं होने देगा, राइट? सो व्हाट इफ यू आर एडिंग इन दिस ब्लड सम एक्सटर्नल फैक्टर्स लाइक टिश्यू फैक्टर्स विच आर रिलीज ड्यूरिंग द इंजरी. So when you are adding tissue factors in the blood which contains even the anticoagulant, it start clotting. So that means that is your extrinsic mechanism. Means there are means apart from clotting factors, there are other factor also which can initiate the clotting process and which can helps in the clotting. So that are your extrinsic mechanism. So first we are going to discuss about the intrinsic mechanism, how blood clotting is going to start. So in intrinsic mechanism of blood clotting, intrinsic mechanism. So you know, after primary plug, what is going to happen? Intrinsic mechanism, there are various factors which are there in the blood, but they are in inactive state. Whenever there is any stimulus, when there is a clotting is required, so these factors will convert from inactive state to active state. So in intrinsic mechanism, there is a factor 12 which is inactive, but due to stimulus it's going to activate into factor active state. A stands for active, so factor 12 active, and in presence of factor 12 is going to activate another factor, that is the factor 11. So factor 11 is going to into the active state. Who is activating this? Factor 12A is activating factor 11 which is inactive into the factor 11 active state, factor 11 active state. Now in the presence of factor 11 there is a factor 9. Factor 9 is going to activate and it's a factor 9A, and in the presence of factor 9A, factor 10 is going to activate into factor 10A. So you can remember these as 12, 9, 12, 12, 11, 9, 10. So first is factor 12, then 11, then 9, then 10. And this factor 10A, in the presence of factor 10A, factor 10A activated is going to convert your prothrombin into thrombin, and this thrombin is going to convert your soluble fibrinogen proteins into insoluble proteins, that are fibrin insoluble proteins. So monomers will be formed, right? Again, what's happening? Factor 12, factor 12A, 11, 11A, 9, 9A, 9A, and here are also other factors, means other substances which is required for these activations, and particularly calcium is also helping. Your calcium's vitamin K is well known coagulants. Vitamin K helps in the coagulations. Vitamin K, calciums, right? So these chemicals are helping to activate factor 10 into factor 10A. So calciums, vitamin K, particularly there is a these factors. What are these factors? These factors are proteins, and particularly these proteins are your glutamic, and glutamics is your beta carboxylated glutamic acids. So when your beta carboxylated glutamic acid, when it's carboxylated, it contains CO negative, right? So how these will be activate each others? They have to bind first and then they will activate. So how it will bind? Calcium is helping because it's CO negative, then there is again CO negative. So calcium is helping like a binder. And where these reactions are happening, these reactions are happening on the surface of platelet. And where are the platelet? Where your platelet primary plug is formed. So where these clotting mechanisms are happening? These are happening on the surfaces of platelets. And surface of the platelet is made up of what? Cell membrane of platelet is made up of what? Made up of phospholipids. So there is a phospholipid, right? On phospholipids, there are your proteins, and these proteins are your like glutamic acids. Glutamic acid is one of the amino acids, right? And this glutamic acid is carboxylated, and these carboxylated bind with calciums, and they are activating one factor to another factors, right? So particularly this fibrinogen is soluble proteins, but by these reactions this fibrin monomers will be formed, monomers. So what will happen? As I have told you, there is a platelet plug, primary platelet plug. There are a lot of platelets which have stopped the bleeding. So now on these platelet plug, what is formed? Fibrin. So fibrins are formed, monomers are formed, right? Now this platelet plug is going to get the strengthened, right? So but these are monomers, so they have to be cross-linked with each other. When they will cross-link to each other, then it will become more stable. So how these will cross-link? So thrombin is going to activate another factors, that is we call thrombin stabilizing factors, TSF, thrombin stabilizing factors, also known as factor 13. So factor 13 is also known as your thrombin stabilizing factors, factor 13. So this factor 13 is what? Its factor 13 is a enzymes which will do the cross linking of these monomers, which are fibrins. So these factor 13, that is your thrombin stabilizing factors, it's going to do the cross linking between these fibrin monomers. So when these will cross link, so this will get lot of strength. So your blood clottings will happen, right? So again I am repeating, what is the intrinsic mechanism? In intrinsic mechanism means blood has its intrinsic properties which helps in the clotting. So if you took the blood in any tubes, it will clot after some time. So that means blood has some intrinsic properties which helps in the clotting. So what are these intrinsic mechanisms? Your factor 12 is going to activate, means when there is no clotting is required, in that cases these all factors are in their inactive state. Whenever there is any stimulus where clotting is required, then these factors is going to convert into the active form, and they are going to activate each other. Factor 12 is going to activate factor 10, factor 10 is going to activate factor 9, factor 9 is going to activate factor 10, and factor 10 is going to convert prothrombin into the thrombin, and then thrombin is going to convert that fibrinogen into the fibrin. Fibrin is insoluble protein, fibrinogen is a soluble protein. So it's a insoluble proteins will be formed, and these monomer will going to deposit, because all these reactions are happening where? These all reactions are happening on the platelet surfaces. So platelet surfaces, there is a phospholipids, there is your factors, these proteins are working. So these will be deposit on the platelet plug, these monomers. But as I said, monomers has to be cross-linked with each other, then it will get more strengthened. So which enzyme is going to do the cross-linking between the monomers? So this thrombin is going to activate other factor that we call thrombin stabilizing factors, or you can call factor 13. This factor 13 is going to cross-link the fibrin with each others, and that clot will get the strengthened. So that is your intrinsic mechanism of blood clotting, right? So this is about your intrinsic mechanisms. Then extrinsic mechanism. Extrinsic mechanism of blood clotting is shorter as compared to intrinsic mechanism. There is a factors, of course there is a tissue factors. Tissue factor is going to activate, right? Tissue factors, and tissue factor is released when there is a endothelial injury, when your blood vessel is going to damage. So tissue factor is also released. So that tissue factor is going to activate factor number 7 to 7A, and this 7A is going to activate your 10 into 10A, and you know factor 10A, then prothrombin, thrombin, and then prothrombin into thrombin, then thrombin, in the presence of thrombin, your fibrinogen into fibrin. That is your extrinsic mechanism. So extrinsic mechanism is smaller than your intrinsic mechanism, right? So ultimately what's going to happen, as I told you, when there is injury, for example, this is your blood vessels, this is your injured area. So what's happening? Platelets are moving in the blood. So their damaged endothelium is going to release one factor that we call von Willebrand factors. Your platelet is going to adhere with the surfaces, that will be called platelet adhesions. Then platelet are going to activate and they are going to release various substances like TXA2, ADP, and making a plug, and this plug we call primary plug. This primary plug is getting the strengthened by the clotting mechanism. So both intrinsic, whether it is intrinsic mechanism or extrinsic mechanism of blood clotting, what it's going to do, both mechanism, they are going to do the formations of fibrin. And what fibrin? Fibrin is your insoluble protein. So monomer is going to deposit on this primary plug, and these fibrin is how they are cross linking with each other with the help of one, there is a thrombin stabilizing factors. Thrombin is going to activate, or you can call it factor 13, one more 13, and that is going to cross link these monomers. So now this will get lot of strength, and that is your mechanism of blood clotting, how blood clotting is happening. So this is all about your blood clotting mechanisms, mechanism of blood clottings. So quickly, if I said what we have discussed, again many times, first whenever any injury, जब भी आपको कोई इंजरी होती है, ब्लड निकलता है तो मीन्स आपके एंडोथेलियल सेल डैमेज हुए, तभी ब्लड निकला, राइट? जब एंडोथेलियल सेल डैमेज हुए तो क्या होगा, सबसे पहले प्लेटलेट वहाँ पे अटैच होना शुरू हो जाएंगे. देयर कैन बी वैसोकंस्ट्रिक्शन, देन देयर इज अ वैसोडाइलेशन मैकेनिज्म एज वेल. सो दिस प्लेटलेट इज गोइंग टू एडहियर एंड देन प्लेटलेट इज गोइंग टू एक्टिवेट, मोर प्लेटलेट इज गोइंग टू एग्रीगेट. तो वो क्या करेंगे, आपके जो खून आपका निकल रहा है, ब्लड निकल रहा है, उसको बंद कर देंगे, राइट? पर मैंने बताया जो जिस वजह से बंद हुआ आपका ब्लड इनिशियली, दैट इज ड्यू टू फॉर्मेशन ऑफ प्राइमरी प्लग. सो प्राइमरी प्लग इज़ वेरी लूज. अगर आपने उसको छेड़ा, खुदेड़ा दोबारा से, तो क्या होगा, दोबारा खून निकलना शुरू हो जाएगा. तो दिस प्राइमरी प्लग इज गेटिंग द स्ट्रेंथन. इसको मजबूती कहाँ से मिल रही है? मजबूती मिल रही है कोगुलेशन से. कोगुलेशन के मैकेनिज्म एक्टिवेट हो रहे हैं. तो जब ये एक्टिवेट हो रहे हैं इंट्रिंसिक और एक्सट्रिंसिक पाथवे, तो फाइनली क्या हो रहा है, वो एक सॉल्युबल प्रोटीन्स जिसका नाम है फाइब्रिनोजन, उसको कन्वर्ट कर रहे हैं फाइब्रिन में, और ये सारे रिएक्शन आपके प्लेटलेट्स के ऊपर हो रहे हैं और उसको स्ट्रेंथन दे रहे हैं, इसको प्राइमरी प्लग को. तो फाइब्रिन आपस में क्रॉस लिंक होंगे एक और एंजाइम से जिसको हम बोलते हैं थ्रोम्बिन स्टेबलाइजिंग फैक्टर और फैक्टर नंबर 13. तो वो उसको क्रॉस लिंक करवा देगा और आपका क्लॉट, आपने देखा होगा जब आपको चोट लगती है एक दिन में वो थोड़ा सा लूज होगा, मीन्स उसको आप ये करोगे तो वो ब्लड दोबारा, पर आप देखोगे 2-3 दिन बाद वो क्लॉट बहुत हार्ड हो जाता है. तो हार्डनेस कौन प्रोवाइड कर रहा है? योर क्लॉटिंग फैक्टर फाइब्रिन. फाइब्रिन जब आपस में क्रॉस लिंकिंग हो रही है तो वो एक्चुअल में हार्डनेस प्रोवाइड कर रहा है, राइट? सो दिस इज अबाउट योर blood clotting, mechanism of blood clottings. Thank you.